Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute and the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back, everyone, to the Genetics Podcast. I'm excited to be here today with Michelle Werner, who's the CEO of Alltrna and CEO partner at Flagship Pioneering. You may have heard Michelle before on episode 139, where we talked about rare disease breakthroughs using tRNA. This is one of the episodes that I really enjoy, where we get to do a little bit of a follow-up with a guest and hear what's changed in the last year or two, and also obviously what's on the horizon for them. So Michelle and the team have published some really, I think, breakthrough data in the space that they've been working on and have continued to push the envelope towards clinical studies in what I think is one of the most interesting ways to target the rare disease population in a way that takes an otherwise very small population and makes it quite a bit bigger, which means we can get treatments to patients who otherwise might not get them. So, Michelle, thank you so much for joining us on part two of the podcast.
Well, it's my pleasure to be here, Patrick. Thanks for having me again.
I would love for people who haven't heard you before, they can obviously always go back to episode 139 and listen, but it'd be great if you could just give a quick overview of Alltrna, what you all focus on, and also your background and motivation to spend so many important years of your career on precision medicines and rare disease.
Sure. So at a very high level, what Alltrna is all about is we're all about tRNA or transfer RNA, hence the name Alltrna. And really that's, we're in the business of unlocking the biology of transfer RNA and turning it into a novel modality for patients. And in the first instance, what we're doing is using tRNAs to read through nonsense mutations. So that's any of the genetic diseases or patients with a genetic diagnosis that stems from a premature termination codon would be kind of reclassified as what we call stop codon disease. And by reading through these premature stop codons with our engineered tRNAs, we can restore the production of a full-length functional protein. And what's super exciting and cool, as you alluded to, Patrick, about the tRNA is that the same tRNA can be used across hundreds, maybe more, different diseases, because we're really targeting the mutations that are common across these diseases, rather than taking the traditional gene by gene or disease by disease approach that really every other genetic modality must take. So it really has this ability to address a much broader set of patients across diseases than we've ever seen before. So we're really excited about that.
Yeah, it kind of blew my mind when I first heard about it because it's almost so simple that it's elegant. Like there's a double-digit number of tRNAs in theory if you restricted to a particular organ system that you could design, right, to potentially treat the vast majority of patients with these nonsense mutations. Could you give an example for the people who aren't thinking about this every day? Like what is and if it's public, you could talk about the first triplet that you're targeting and how that works, just to bring this home to like when a patient's DNA is sequenced and it looks like this, then they might be eligible for this kind of tRNA therapy.
Yeah. So first of all, in general, about 10% of all genetic diagnoses stem from a premature termination codon or a nonsense mutation. And that is where we would say they would have stop codon disease. And there are actually 19 different ways in which a premature termination codon can occur. And as you know, this is where you have a code for an amino acid codes for a stop instead. And there are 19 different possible ways in which that occur. However, what we know is that actually they're not all distributed equally, those 19. In fact, there are two that are the most prevalent. And that's the arginine to TGA stop mutation and the glutamine to TAG stop mutation. And they represent these two about 50% of all patients that have
Wow.
stop codon disease, right? So we, of course, are focused mostly on those two. So our first program is that arginine to stop mutation. That's where we've disclosed our data for our development candidate, AP003. But we have a second program following just behind it that's focused on the GlynTag mutation, which is the second most prevalent of them all.
Amazing. I want to come back to the tRNA space in general, because as far as I'm aware, there's no approved product with using tRNAs, and you're the first or amongst the first to make it as far as you have. Before we go into that, though, I want to just talk about the concept of basket trials, because I think it's really important to think about how this ultimately gets to patients. Could you explain that concept for people who aren't familiar with it, and talk about how it applies in this situation?
Yeah, so a basket trial design is not a novel clinical development strategy. In fact, it's actually used quite prolifically in the oncology space and has led to the approval of, I'm going to say, around 7 or 8 different approved medicines by the FDA using a basket trial approach. And what that design looks like is patients who technically have different diseases. And so in oncology, it might be a lung cancer patient and a breast cancer patient, for example. But they all have the same genetic mutation that is the driver behind that disease. So, for example, EGFR can be consistent across multiple different tumor types. And the same is actually true in rare diseases, can be done. And so we've seen basket trials who take those patients that have technically a different disease, but those patients are all identified by that common mutation, and they're treated with a single modality, a single therapeutic in that clinical trial. You measure the same or similar outcome across those patients, despite the fact that they have technically a different disease. And that can be a registrational strategy. So as I mentioned, very used prolifically in the oncology space, so there's lots of precedence for it. In rare diseases, it has been used less often, and that's really because most of really the genetic approaches, as we talked about earlier, must take this very gene-specific, disease-specific, or protein-specific type strategy. And so grouping patients that have different types of diseases doesn't really make sense. But when you're using a modality that is focused on the common mutations across those different diseases, it does allow us to be able to use this basket trial design in a way that we haven't really seen it before. It's been used maybe with some small molecules that can be used across different diseases in the rare disease setting. But I do think maybe the tRNA might be the first basket trial strategy or genetic modality for rare genetic diseses. So that's pretty
Yes.
cool.
That's amazing. So you, since we last talked, you published some compelling data where you've shown a significant milestone, first in mice, that's going to take you on to hopefully humans in the future. I'd love if you could talk through what you found there, and then from that, I want to talk about some of the bottlenecks, because this is when you're doing something the first time, you're crashing through a few different walls to get there, and proving that it works in mice is one of those walls for sure. So I'd love to hear about that one first.
Absolutely. And, we fundamentally believe that demonstrating preclinical proof of concept, meaning, what do these tRNAs look like in an in vivo disease mouse model, really has to be an indicator or bellwether for how that might translate when we move into humans, which of course is what we're the direction that we're heading to next. So we really felt it was important for us to be able to demonstrate in an in vivo setting that these tRNAs can do what we think they can do, which is read through these nonsense mutations and restore protein rescue and restore wild type protein production. So the data that we presented in December of 2024, we're really, really excited about. So we presented protein restoration in 2 different disease models, methylmalonic acidemia, or MMA, and phenylketonuria or PKU. Now, both of these mouse models have an arginine to stop mutation. And so we've been able to use our tRNAs that are directed for these arginine to read through these arginine to stop premature termination codons in these experiments. So what we were able to demonstrate in the MMA model, so methylmalonic acidemia, is that we were able to restore the human M-mut protein to levels of around 25% of the wild type at day 4. And we also saw protein restoration at 14 days, and I'll come back to that in a second. I will share with you, Patrick, that actually we've continued to do work on this particular model and analyzing some of these results. And actually, we believe now that with the tRNA that we've used in that experiment, that we're actually able to restore almost 50% of the human M-mut protein. So a real significant amount. Now, to put that into perspective in terms of the disease, so MMA, a very serious life-threatening condition that is, patients unfortunately exhibit symptoms at a very young age in infancy, often within days or just a few weeks of birth. And when we speak to clinicians, key opinion leaders who are actively treating MMA patients, by their estimations, they believe that really 1 or 2% of the human M-mut protein is enough to really significantly ameliorate that patient's symptoms. And that's because these patients, unfortunately, really can't produce any of the protein on their own. So when we see nearly 50% at 4 days, and actually we saw about 10% at day 14, we're well above that 1 or 2% sort of like minimum that clinicians think would be required. So we were really, really, really excited about these data and very encouraged by what that might mean and how that might translate then to humans. Now, the other piece of data that we also shared, as I mentioned, is in a second disease model, and that's phenylketonuria or PKU, again with the same arginine to stop nonsense mutation. And in this experiment, we showed a rescue of the PAH protein to 7% of the wild type levels. That was within 72 hours. And that, again, to put it into perspective of what clinicians are expecting to see, they think that about 3% is what patients would need in order to be able to ameliorate their condition. Now, that 7% of PAH protein can actually be measured. We can measure a functional readout in this particular model because it is this particular animal model, not only is not just a transgenic mouse model, which is what the MMA model is, but it's actually a functional model, which means we can also measure the biomarkers associated with that disease.
Oh, yeah.
In PKU, the biomarker that is measured clinically is phenylalanine, and it's a registrational endpoint. Actually, it's used to approve several medicines by the FDA. And that 7% protein restoration translated into a 76% reduction in phenylalanine levels, which is actually quite massive. And so we're really excited about those data too.
Yeah, that's amazing. When you set out to do this, was that what you were expecting, or did you have any hint of what it might look like? Because this is a nervous moment for the CEO to wait for this data to come out and see what it looks like, right?
I mean, yeah, trust me, I was anxiously awaiting these data, especially because these PKU mice are actually quite fragile because they have the disease itself.
Right.
And so it wasn't very easy actually to keep those pups alive for long enough in order to be able to receive this treatment. So that was even encouraging for us to be able to get to this point of having these data. And so, I mean, honestly, what we were hoping to see is a demonstration of read-through of the premature termination codon, that we'd like to see protein restoration at levels above what clinicians have asked us. I never anticipated seeing a 76% reduction in phenylalanine levels. So that was really, truly meaningful data for us. And having levels that, a reduction at that high of a level really makes me feel very optimistic about the translation of these data from species to species. And of course, that means from mouse to human as a next step.
When you so as you start to think about translating this into humans, some of the bottlenecks that I'm aware of when it comes to tRNA, or some of the challenges, I guess, is delivery is one big one. How do you get it to the right organ system? Maybe we can start with that one. What have you learned about delivery? And I think you're going to start with the liver, but you could apply this to any organ system over time. So I'm curious to hear your thoughts on the delivery space in general. It comes up a lot on this podcast of people saying we have okay tools for the liver, but everywhere else is in
Still a
need of
work
some
in
help.
progress.
Yeah.
Yeah, that's exactly right, Patrick. So yes, our decision to formulate in the first instance with a lipid nanoparticle was a specific and strategic decision, and it was really a de-risking decision. So, of course, with any oligonucleotide, it requires the ability to deliver to the site of action. And in LNPs now, we know, have been in millions of people around the world. We know exactly how well they get to the hepatocytes, what their safety profile looks like. And so, being able to formulate with an LNP is something that can be, readily done and allow us to really de-risk that delivery in such a way so that we can focus on the payload, and really keeping in mind nobody's ever received a tRNA before
Right.
as a therapeutic. And so it's a completely novel space. And for us, it was really about how do we make sure we can focus on the payload and what it's doing, with having a lot of reassurance about what the delivery vehicle itself can do and is doing with this particular payload and with these diseases. Now, the good news is that when it comes to liver diseases, there are roughly about 400-ish rare genetic liver diseases that may be amenable to a tRNA therapeutic as a technology. And even with an LNP formulation, given this idea about being able to use a single one of our engineered tRNAs across many different diseases, we still have the opportunity to prosecute many different diseases at a time using a basket trial strategy, even with the sort of specific focus on liver in the first instance, given the LNP delivery. What I will say is that a tRNA can actually be amenable to many different types of delivery vehicles. And, one other type of delivery vehicle that's obviously used quite commonly for genetic diseases are AAVs. And, theoretically, we can use a tRNA in an AAV formulation as well to tackle certain diseases. And that might be a strategy that either we or other companies might be using. But we also think that it doesn't have to be just LNPs and AAVs, that a lot of the work that we're actually doing from a tRNA design perspective, which is not just focusing on how to optimize the sequence of the roughly 75 bases that make up a tRNA, but then also decorating it with different chemical modifications to enhance its clinical properties, such as their activity and potency, which is why we see such encouraging protein restoration data in vivo. That the work that we're doing on design around chemical mods in particular actually help us understand how we might be able to use a tRNA with different conjugations and linkers, and maybe in the future be able to combine our tRNAs with some other technologies that are actually advancing in the field, that could perhaps deliver to the CNS or muscle or the
Yeah.
lungs, for example. And I'm really excited about the possibility of that too.
Yeah,
But
that's really interesting. Are
we do
there
have to walk before we can run. So let's get into
yes.
it first and show that it works.
It is a gift and a curse of having such a broad platform, right? You can future cast on a lot of these things, but you also have to be very, very focused around what you can do in the near term, right?
100%.
Are there any of those like, because I went down a rabbit hole recently learning about there's the chemical modifications you talked about. There's also RNA, like I think they call it RNA origami. There's like a folding. You can structure them in a way that they fold into a kind of vesicle for the RNA to go in. There's obviously nanoparticles. There's viral vectors. Are there particular strands of this that you think are interesting, that people are more aware of AAV, and some of these others that I think will continue to be very important? But I'm curious about any of the more esoteric ones that you've seen that could be interesting.
I'm not so sure, but I will say, and I'm very focused on, I'm very focused on what we're doing here at Alltrna, but I think also because of my personal circumstances, Patrick, and I know you and I have spoken before in the past about, I have a son with Duchenne muscular dystrophy, so I pay a lot of attention to
Yeah.
the advancements that are happening in that space. And even when you look at just in the Duchenne or muscle disease arenas, peptide conjugations, antibody conjugations,
Yeah.
all of these are actually like the data are looking really encouraging. And so I think delivery to muscle is, it's within sight. And I do think that a tRNA might be able to, latch on to some of
Yes.
different technologies in the future. And so yeah, so those are things that I'm, really curious about and sort of willing on and hoping that they're successful too.
Something else that I read was that it's a relatively unstable molecule, and I guess there's a lot of competition for other tRNAs. In spite of all that, you've been able to show what looks like effective dosing in the mouse model. Is that a big challenge, or how do you navigate that? Because it's probably a messy world of lots of tRNAs competing for doing the job, right?
Yes and no, actually. So first of all, what I would say is, I think we should keep in mind that the tRNA is in its preferred state in a folded structure, right? So oftentimes
Mm,
we see the tRNA in our textbooks and they have this kind of clover leaf-like design,
yeah,
two-dimensional flat, design. But actually it really folds into this almost L-shaped structure, which is quite frankly a lot more stable than, let's say, an mRNA, which is more labile in nature. And
yeah,
so there's a big difference between the tRNA in that state versus other types of RNA. And so I think that's, something that should be noted. So, I mean, yeah, so we should keep in mind that the tRNA is actually the most frequent RNA in the system. So, yes,
right,
there
yeah,
are a lot of tRNAs around. However, what we also should keep in mind is that there are no natural tRNAs that can read premature termination codons. And that is why we get
oh
truncated
yeah,
protein, because
interesting,
their natural tRNAs don't exist. So when the
yeah,
ribosome is doing its job and it's sampling, for all the tRNAs in the environment, that's what happens. It just samples until it finds the right tRNA. And because it doesn't have a
mm,
natural tRNA, that can read the premature termination codons. That's why the protein translation stops. So the
right, so
ribosome
just stalls, yeah,
releases, right? And the protein is released. It then has that truncated or dysfunctional protein that
yeah,
is produced at the end of the day. And so there actually isn't a lot of competition that's happening when we reintroduce an engineered tRNA.
yeah,
So the engineered tRNAs are actually recognized by the ribosome endogenously. We actually are delivering tRNAs that are uncharged. Once they're recognized by the ribosome, they get the natural modifications that would be put on any tRNA. So the engineered tRNA receives the natural modifications as well, and then gets charged endogenously. So it knows exactly which amino acid it's being coded for. And so when that sampling is occurring, that when it gets to the premature termination codon, instead of sampling and not finding the right tRNA and therefore releasing, it samples and then finds the engineered tRNA
yeah.
quite easily. So that's at least what we believe is occurring based on both the in vitro and the in vivo data that we have seen to date. So it sounds like there would be a lot of competition, but there actually isn't. So
Yeah,
that's
make
why
total
we have
sense.
biology works in our favor there.
Yeah, that's very cool. You had me thinking as well about future directions of this. Again, not today. We have things to focus on, but you could start to introduce engineered tRNAs that look at missense or frame shift or other kinds of changes, right? Is there anything stopping us from that from a cell biology perspective?
No, I definitely think that there are other possible applications beyond nonsense mutations. Again, it's the walk before we can run, and
Yeah,
nonsense mutations are a little bit more straightforward, perhaps, than some of these other mutations. But I do believe that it's possible, and so certainly in the future, once we can demonstrate the clinical proof of concept with the modality, I think it makes sense for us to continue to explore those applications much more broadly.
yeah, super interesting. If we dive a little bit into the basket trial and innovative trial design more broadly, it's an area that I'm super interested in. How do you think about like with the basket trial paradigm you described, you could have two diseases like PKU and MMA, or you could have 20 disease, or you could have 400. You said there are 400 genetic liver diseases, but scaling up creates some complexity and coordinating costs and lots of challenges. How do you think about striking that balance between the breadth and the depth, I guess, of a program like that?
Yeah, so we thought a lot about this actually. And yes, so 400 diseases sounds like a ridiculously complex basket and not one that would be a clinical trial I would want to run.
Yeah.
We also wanted the data to be informative, right? And so what we hope to be able to demonstrate is that across a certain number of diseases, that we're demonstrating the ability of our tRNAs to read through those the same premature stop codon and able to restore protein levels or the relevant biomarker, I should say, have an impact on the biomarker clinically. That's what we would be measuring. And if we're able to demonstrate that across a subset of diseases, then I think we, prove the idea that, the
Yeah.
universal way in which a tRNA can be used across a number of different diseases, the whole premise behind what we're all about, has been realized. Now, when we think about the initial basket, a lot of the things that we considered were, first and foremost, diseases that we thought were, where we see the prevalence of the nonsense mutations that would make sense, where we believe that the underlying biology is amenable to a tRNA type of technology. And actually, we're looking at diseases where there are biomarkers that we believe that can be measured in order to be able to understand or to demonstrate if there's read through of the premature termination codon. And we're actually choosing diseases that have a common pathophysiology because that just makes things a little bit simpler. That's at
Right.
least for the first basket. Now, maybe in the future, once we demonstrate that, we can go much more broadly to diseases that are far more disparate in nature. But we, again, wanted to be able to have a subset of diseases where we felt confident and comfortable that we have the ability to demonstrate what we think can be achieved. So having seen what we've seen in methylmalonic acidemia in a disease mouse model, the same thing in phenylketonuria in a disease mouse model, these gives us reason to believe that, well, we've seen it twice in two different disease models. Biology is very highly conserved across these diseases. The biology of the underlying mechanism of action of a tRNA is also really highly conserved, that it's very logical to assume that you'll see something similar across many diseases. And so that's how we're designing our basket to try to prove that concept.
Yeah, amazing. I think you have some other thoughts around extending the basket trial concept, right? We can talk about adaptive trial design maybe and how you don't necessarily just set the plan in stone at the beginning, but you actually adapt based on predetermined data locks at certain points in the process. It would be great if you could expand a little bit on how you think about that paradigm.
Yeah, so we're seeing a lot of adaptive clinical trial designs moving, let's say, from a phase one right into a phase two or a phase two right into a phase three, for example, quite commonly, especially in the rare genetic disease setting. And I think a lot of that has to do with the somewhat limited patient populations and trying to make the most of those populations and those data and informing the decisions of what happens next in a pretty real-time type scenario. And so we think that those strategies can be achieved here as well. So we may move right from a small, sort of very highly defined number of diseases in a basket. And once we look at those data, if the data seem encouraging, and of course, not just from a pharmacodynamic or effectiveness perspective, but also a safety perspective as well, which we'll be looking extremely closely at, of course, then we might be able to move into a broader basket of diseases beyond just the initial subset of the diseases too. So, and I think that that's a strategy that is, particularly amenable again for a tRNA, where if you prove the biology works in a clinical setting, that it makes sense to broaden the numbers of diseases, because keep in mind that most of these diseases don't have many or any approved options, and many of these patients are underserved with what's available for them today, that it would make sense to broaden the possibility of including those patients and those with those different diseases in a broader clinical setting.
Yeah, how a lot of this is going to be new to regulators, but when there's high unmet patient need, there generally doors are very open to hear creative and interesting ideas. If you're able to talk a little bit about it at a high level, what has the feedback been from the EMA, the FDA, whichever groups you've been um, coordinating with to get feedback on this approach? Because it's like I said, it's novel, but it's hitting a very important unmet need.
Yeah, so I definitely think so. Taking a step back, I think when you have a new modality, having early and active engagement with health authorities is generally a good strategy, and that's certainly one that we've embraced here at Alltrna. So I'd say first and foremost, that's like a critical component of what we've been doing. Secondly, I would just say some of these data and statistics around rare diseases, which we've talked about before in the past, roughly around 10,000-ish different genetic diseases. And if we're tackling them one at a time, health authorities also realize that it's going to be forever. Many patients, unfortunately, will die before we have approved medicines for each of those conditions. So they're actually already starting to take a narrative of trying to find these many diseases at a time strategy in order to be able to address that problem of too many diseases and not enough time. So I think that's something, the environment is really ripe for this type of technology and this type of development strategy to take hold. The conversations that we've had to date are really encouraging, right? Keeping in mind this is a brand new modality and there's a lot of work that we're going to need to do and ongoing conversations that will be required. But because there's a lot of interest in a tRNA and the possibility of a tRNA not just tackling the most common of the rare diseases, which is typically where we have the approved medicines and will likely continue to only have the approved medicines because those populations warrant that, individual disease development strategy, that, the ability to really bring in patients that have these ultra rare diseases who may not otherwise actually have their own clinical trial at all in a patient's lifetime, maybe in generations of patients' lifetimes. That's something that I believe that there's quite a bit of interest in helping us figure out. Now, I do think that they're going to ask us to see, the appropriate preclinical data sets. And so these MMA data, these PKU data will be amongst those preclinical data sets that we think are going to be necessary to justify the clinical approach that we're proposing. And also keeping in mind, Patrick, that as you know, most of these rare genetic diseases address a pediatric population, right? I mean, if you think holistically, 80% of genetic or rare diseases affect children under the age of 5. So it's a lot of these individuals are young. So we want to make sure that we're doing the appropriate work to demonstrate as well as we can the toxicology package that's required for health authorities to go into populations that are younger in age, but then also to be able to really demonstrate our ability to be used broadly across diseases. So I think there's certainly work on our part that we're already well underway to make happen, but a lot of it will also happen in conversations with health authorities and getting them comfortable and familiar with this approach, and not, to really realize that we're not, we're not taking unnecessary risks here, right? We're doing the hard work that's necessary, but that also putting into the context of, patients are going to be missing out if we can't figure this out, and creating a partnership type approach that we can work through some of these challenges together.
Yeah, I mean that's a challenging tightrope to walk sometimes where you want to move as fast as possible because patients are waiting, but also if you move too fast and you screw up, then you throw the baby out with the bathwater, right? And you never get there. How do you have any frameworks? You've been leading Alltrna for almost 3 years now. I think it'll be 3 years in April. What have you learned during that time about how to balance that tightrope and lead a cross-functional team of people more broadly?
No, I think that honestly, it really comes down to us being able to communicate and to demonstrate what we're doing with conviction, but also credibility at the same time. And that's not just, certainly I think that holds in our conversations externally with a number of different stakeholders, including the regulators. but I also think that's true internally as well. So, I mean, we're a company of around 65 alternators, right? Employees right now. And we're all in this because we know we're doing exciting science that could have a very profound impact on patients that's unlike anything else that exists today. But, we also need to remind ourselves that this isn't just crazy talk here, right? That there are reasons to believe why why this can be true. And with every experiment that we're doing internally, that we're building that credibility for ourselves, and that credibility based on data also helps build the confidence that what we think can be done. And so I think that's really one of the beautiful things about being in a company like this at our stage. Every day matters and every experiment matters, and every experiment helps us take one step forward towards our goal. And that's a super cool and exciting place to be.
I've heard you use that phrase reasons to believe a couple times. Is that like a framework that you have or a mantra that you have how you think about it? It feels like a scientifically driven way to run a company in a way you creating this ladder of evidence in a way. But I'd love to hear a little bit more about that if that's a way that you think about operating more broadly.
Yeah, sure. And I would say it's not just an Alltrna thing, but actually, I think if sort of take a step back, I mean, Alltrna is a flagship pioneering company, which is the, sort of the larger parent, sort of venture creation type, sort of brain power behind many, many, many different companies doing some incredibly cool things right now. Moderna, of course, being one of them, which I'm sure everybody is familiar with. But even when you think about the framework that Flagship uses to do company creation, it starts with a fundamentally, different scientific what if question. And the
Yeah.
what if question for Alltrna is what if you can use a tRNA to address genetic mutations. And so I don't know if anybody has ever asked that question before Flagship asked that question. And you start with this kind of big idea with the what if question. And then the next step, I'd say, is really about, okay, well, do some reason to believe experiments to see, is there feasibility there? Is there
Yeah.
validity to that idea? And that's exactly how Alltrna got started back in 2018. So we asked the what if, and then the initial, founding scientific team started to do those, fundamental reason to believe experiments. Can you deliver a tRNA to the site of action? Can we quantify them? Can we differentiate an engineered tRNA from an endogenous tRNA? Well, can we actually engineer them at all, right? Is there a way to chemically synthesize these molecules in a way that, because it hasn't been done before. So all of those were the sort of initial steps that were taken right at the very beginning of the company. And now we're still doing those reason to believe experiments all the time still. They're just
Yeah.
different reason to believe experiments. And so, yeah, I do think we have to continue to ask those questions. And even now that we're getting closer to the clinic with our first development candidate, that will be the big question, right? So does what we see in mice translate to humans? Is there reason to believe that that is true? Can it be safely tolerated in a clinical setting? Those are going to be our reasons to believe in those initial clinical trial or trials that we do before we kind of expand to, build out to the sort of the way in which we think this platform can.
Yeah, I really like that as almost like a companion framing to de-risking. We talk about de-risking a lot in this industry, but that can lead you to you can de-risk so heavily that you never do anything that is a big swing. What is different about this is you're saying, let's pick something that we know if we could achieve it, it would be amazing. And can we draw at every step of the way, can we draw a thread to the next step? Do we still have a reason to believe that we could get to this very exciting future? So there's some de-risking involved in it, but it's really about, do we believe that we can get to this very exciting thing in the end versus let's just continually de-risk until we know that whatever we do, it's going to be successful no matter how incremental it might be.
That's exactly right, Patrick.
As we wrap up here, I was also just curious to get your perspective on small biotech versus large pharma. You spent a number of years at AstraZeneca, as well as Novartis, if my memory is correct.
And
And
BMS, yeah.
you were leading major franchise teams in hematology and in solid tumors in both those places. What have you carried over from large pharma to biotech that you think has served you well, and what are the things that you had to almost unlearn or get out of your system?
Yeah. So first of all, I would say that, I cherish my experience in large pharma. I'd say that the 20-ish years that I was there in a variety of different roles, I had the opportunity to have a breadth of, different experiences in many different countries and many different functions and on different medicines and different disease areas. It was great training ground for what I'm doing now, and I will, I will never regret my experiences there. It's been very positive and beneficial for me, personally and professionally. What I would say is what I take from those experiences is really how to understand how to navigate complexity, how to navigate uncertainty, how to navigate different stakeholders and manage different stakeholders, and to work within a matrix setting. Um, and even though Alltrna is a fundamentally different and smaller organization, there's still complexity and uncertainty in the work that we're doing. There's a lot of stakeholders and matrices that we need to interact with, not just internally, but also externally as well. And those are all things that I would say, those leadership experiences that I've received before definitely are playing out here. I certainly would say that some of the things that I've had to unlearn is that oftentimes when you're in a larger company, there's a lower tolerance for risk in some ways than, of course, in a smaller company where, as you said earlier, Patrick, you're swinging big, right? You're swinging for the fences on things that have never been done before. And there are some times where I will say I have to check my risk tolerance a little
Right.
bit. And and so and I would say I've also been more having a higher propensity for risk or higher tolerance for risk than might traditionally be the case in large pharma, but it's
Right.
definitely something that I would still say I have to, to check sometimes. And so that's definitely something that I would say is, top of mind. But a lot of the, how you lead people and like, yeah, like you said, I've led big teams, big organization, hundreds of people, large P&Ls. It's very different in a small, nimble organization like this, but leadership is leadership, and the translation of why we're doing what we're doing and the impact that it can have is the same. Whether or not you're leading a team of, 500 or 5,000 people or 50 people. But really putting that into perspective of the why, I think is something that I've learned a lot from my experience in big pharma, but also with my personal circumstances and connection to the genetic disease space. And I think that leadership component has translated very nicely from one environment to this one.
Absolutely. How, as we wrap up here, how can people be helpful to you? Are you hiring? Are you looking for partners? What would be most useful?
Well, we're definitely always interested in talking to people who maybe like are as curious and interested in our technology as we are. Certainly when it comes to strategic partners, pharma partners, I think that because we've have a platform that has the ability to expand in so many dimensions, being able to bring experts on board who have been in the rare disease space, who really understand the ins and outs of development could be an asset to us if we want to go down that road. And so we'd be, open to having that. But I would say just in general, kind of, continuing to push the envelope about the sense of urgency around rare diseases, the need for continued regulatory flexibility, the continued importance of doing genetic testing as baseline standard of care, no matter what that your diagnosis is. It should be mandatory, I think. All of those things I think are going to be really just beneficial for the industry as a whole, and Alltrna is no different than that.
Yeah. Do you have any thoughts on the current state of rare disease when it comes to the new incoming administration? Obviously, we saw that the priority review voucher program wasn't renewed at the end of the year. I'm just interested. Like I'm watching very closely to try to understand what the impact is on our space. I'm curious if you have any thoughts or perspective on that.
Yes, so I would certainly be very clear in my position. I think the pediatric priority review voucher or pediatric rare disease priority review voucher really is an important program. And I know it's sunsetted at the end of the year, but I'm optimistic given the bipartisan support that we had for it, that it will continue, it will emerge again. And be an incentive program. I think that's crucial because we really need to be able to keep those lines open for companies like ourselves and many, many others to continue to invest in these space because patients need it at the end of the day. They're waiting for these innovations, and these types of programs really help with that. But I would say is, regardless of the change of administration, the rare diseases transcend any administration. The work that we do at Alltrna can and must transcend any administration. And regardless of what's happening and the decisions that are made, is that we need to stay focused and we need to make things happen for these populations. Patients are waiting, patients deserve better, and we need to get to work. So that's what we're doing.
Couldn't agree more. Well, thank you, Michelle. This was really great. Congratulations to you and the whole team on all the progress. I'm excited to keep watching and seeing all the amazing impact that you'll have.
Yeah. Well, thanks so much, Patrick. It's been great, and I appreciate you inviting me again.
My pleasure. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.